Hello, sorry about that. There was a little glitch with the iPad. Hopefully that got corrected and my O got removed. But we're talking about elastin, and the point I was trying to make was that the eye of round or semitendinosus, or also called the hamstring muscle, is higher in elastin. That's why it's so important in movement, so it needs that extra strength. And that's why the eye of round or semitendinosus is a tough muscle and hard to control the tenderness of it. The third fiber is reticulin. Reticulin is composed of very small fibers. They form a delicate network, kind of like a little spider web. And as we have already talked about there, in around cells, blood vessels, neural structures, and epithelium. And they appear earlier in embryonic development. But they weren't in the pink picture, were they? Meaning that they're not really a major component of tenderness in young animals. They're important in development. Now, we talked about collagen solubility, and I've given you 3 papers that go along with this lecture. I've already talked about the Purslow paper in 2004. You know, it's a very good review paper. It talks about paramecium. We're going to see some more of the information from the Purslow paper. But you also have a Cross et al. 1973 paper. This paper was published by Dr. Cross as a component of his dissertation. If those of you don't know who Dr. Cross is, he's our former department head, and he is still here on faculty. He was actually my co-advisor on my PhD, and I did my work at Clay Center at the U.S. Meat Animal Research Center with him before he came here to Texas A&M. But in his dissertation work, he found that total collagen and elastin were not related to tenderness, but percent soluble collagen was found to be significantly related. And in this paper, there's a lot of information in this paper. It's not that long of a paper. And he had old and young animals. He had 3 age groups. And they did sensory panel work on them. And they did attributes including juiciness, tenderness, muscle fiber tenderness, amount of connective tissue, and then they did shear force. Table 1 is the relationships between tenderness and age and shear force, and the attributes across age groups. I don't really want you to concentrate on that table that much, or looking at table 2, which is the simple correlation coefficients between certain chemical components. What I want you to see is that in table 3 of this paper, the first thing he had to convince me of when I read this paper is that he had age groups that changed in tenderness. And so in table 3, he has the shear force values for the 3 age groups and their standard deviation. This is in the days when you did standard deviation, not standard errors. And you can see that as the animals got older, they got tougher. Some of the things that were changing as they got tougher, if you look at the adjusted soluble collagen, which is partway down, it's like 5th from the end as a component, and what the adjustment was there was adjusting it for elastin. So what they did is the insoluble portion, remember I talked about how you centrifuge it down and you separate soluble and insoluble. In the insoluble portion is some elastin, and so there's a slight different conversion of hydroxyproline amount to collagen to account for elastin. That was a big thing that he did in this paper, but you can see that that is the parameter that followed that change in shear force to the greatest degree. The other thing, then he goes through and presents some information on different muscles, because they also did the semimembranosus, which is the inside round, the semitendinosus, the eye of round, the biceps femoris, which is the outside round, the rectus femoris, and then the longissimus dorsi. and they broke that down into sensory attributes within age group for those attributes. But the one thing that they found when they tried to predict tenderness was that collagen solubility was most highly related to shear force, and that's kind of the guts of this paper, other than the differences in conversion of hydroxyproline in the soluble versus insoluble. Because up to that point, we didn't really think about collagen solubility as being that important. Everybody says total collagen, and Cross was able to show us that we needed to pay attention to collagen solubility, and that differences between animals and tenderness was related to collagen solubility. So that was very important. One of the things that Purslow started helping us to understand, I love these pictures, and you've seen this picture before. That's the epimecium and the paramecium, and then this is a magnification of the endomecium. Isn't it cute? Looks like a little honeycomb. You can see where those individual cells are. But what I also like about this is look at the paramecium. It shows that the structure is different. And we talked about how ground substance contains glycoproteins and fibrin and laminin to help connect. And look, you can see that there's some attachment going on there. This is a blow up of the endomecium. See, doesn't that look like my Chinese finger puppet? See how it's interwoven around there? Because the muscle fibers have been taken out here. I think that's just an amazing picture to help us understand how endomecium and paramecium differ in their structure. And these are some slides. These slides aren't, can't remember if they're in this paper or not. I guess the previous slide was in the paper. Peter, yeah, they are from this paper, but Peter gave me his colored pictures of this. So the Purslow paper kind of reviews most of this, and it is a very good review up to this point in time. But what I will never forget seeing these two pictures. This is paramecium. There's a muscle bundle. Look at the connection of the paramecium to other paramecium. See that attachment? This picture also shows, here's paramecium, and they pulled it apart a little bit so you can see the fuzzies in there, and that's connection. And what Peter is trying to say in this paper is that a lot of it is total amount of paramecium. So this slide right here, again, these are different slides of different muscles showing you this is the pectoralis, this is the sternocephalicus, whatever, I'm sorry, and this is the rhomboideus. And you can see that there's different amounts of paramecium, and that paramecium really is contributing. Here are different muscles right here, and this is their paramecial connective tissue, and this is their endomecial connective tissue. So you can see that paramecium is, as a percent of dry matter, on these tougher muscles, it's a higher percentage of the total dry matter, and that while some of them have, like especially with the extensor carpi radialis, it has higher endomecium, but the paramecium is obviously contributing to most of the effect of tenderness. And he uses these 10-to-1 versus 3-to-1 ratios. So what is going on there? Remember that transmembrane plaque, clear back in the ultrastructure? And we talked about ground substance, and we talked about laminin. Here's laminin. So here is the sarcolemma, and this is that transmembrane plaque. And you see some things going on here. There's actin, and there's laminin. And up here is the basal lamina of the connective tissue, and it's showing that interrelationship. so the trans cell membrane linkages by dystroglycan, which is the muscle specific, and more generally by integrins, connect to the extracellular matrix. These are integrins right here, so laminin integrins, and how this is that basal membrane, this is the sarcoplasm. Remember the alpha and beta integrins that were sticking up? They also help to bind this extracellular matrix along with laminin. And so what we were really looking at, those connections, Peter is talking about how these two proteins are involved. And I just love all of this, all this. I think this helped me to understand it. This is laminin again, and how that transmembrane structures provide mechanical linkages for cell matrix signaling. And that Peter talks about how in growth that is happening, that's in that paper. I'm not going to go through that because that's for later on. But what I want you to understand is that connective tissue is playing a pretty big role in growth and development, and helping to provide that structure, and attachment is an important thing. And also the endomysium is very important, even though it doesn't contribute solely to tenderness, but it helps to keep fibers tightly and registered to give us our grain. So depending on how the muscle fibers are oriented has a big effect on tenderness. And as meat scientists, our one rule is that you always cut across the grain. Because when you cook, connective tissue is going to shrink. And if you cut it with the grain, that connective tissue is going to shrink this way, and it's going to be tougher. But if you cut it across the grain, you cut the connective tissue, and as it shrinks, it's not going to have the same impact. It's not going to pull everything together. And that's what this is trying to show you, is that during contraction, these same lines, look at how they change, and how the density of the connective tissue would change. So paramecium, Purslow is talking about endomysium. It's going to contribute to grain and helping fibers to hold together. It also endomysium helps us to find the grain and those muscle bundles we want to cut across it. But paramecium is the one across muscles that varies the most, and varies more by the functionality of that muscle. And in the internal division of muscles by paramecium, that allows the slippage of the fascicles past each other as the muscles change shape. So he's trying to show that down there. And that there's one thing you probably don't think about. Some of you may not have had a chance or taken a class with different muscles, and the anatomy class that Dr. Griffin teaches, but these are the 6 basic grain patterns of muscle fibers in muscles, the muscles that we deal with beef and lamb and pork. And these are the easiest ones because the muscle grains run the long distance. That'd be the semitendinosus. And then we have some that are going to be kind of circular, oval, and then we have those that fan out, those that run at an angle. This is the longissimus dorsi muscle. This would be the gluteus medius muscle. Some of these include the inside round,